specific for being next to the area that you need to know the sequence of. So you need to know some part of your template strand. You need to know the sequence in some part of your template strand to be able to design an oligo that's going to bind to it, which DNA polymerase can then extend one base at a time. And the other particular that you need to know for sequencing is as each base in the template strand is added to by DNA polymerase. If there's a G here, it's going to add a C. And if there's another G, it's going to add another C. And if there's a C here, it's going to add a G. If there's a T, it's going to add an A. You know how DNA works. So what the DNA polymerase will come in and do, it will add nucleotides. And so the basis of Sanger dideoxy sequencing is that some of the bases that are added specific to each reaction are dideoxy nucleotides. Let's get this to focus. There we are. So in addition to regular nucleotides in sequencing, some of them are dideoxy nucleotides. So there's no hydroxyl here, no hydroxyl here. Remember, RNA will have both, DNA will have one dideoxy nucleotide. There will be no hydroxyl group on either one. With no hydroxyl group, the polymerase has no place to add that incoming phosphate for the backbone of the DNA. So for some of you, this is review. For some of you, this is maybe not quite so much review. So Sanger dideoxy sequencing uses dideoxy nucleotides, G's, A's, T's, and C's, whatever base you're looking at the time. And there's a certain probability that you're going to have each one bind in an addition reaction with the polymerase. Okay, so now I think that's probably most of what you need to be able to understand next-gen sequencing. So next-gen sequencing is kind of like a takeoff on that method. And I did post for you an announcement giving a link to a very nice review. It sort of steps you through a bunch of slides. So if I'm talking too fast for you, if you're like, wait, what did she say again? I want to review that again, particularly for the people who have not yet had basic sequencing and maybe think they're a little bit lost when watching this. You can always go back and go to that link I posted and see that review of how next-gen sequencing works. Okay, so they use the same basic deoxy and dideoxy nucleotides, but what is different in sequencing is, again, they have particular fluorescent additions added to the bases, and those fluorescent additions can be cleaved off with an enzyme. So they can be cleaved off, and then the next base can be added normally in a sequence. So when you are doing sequencing, did I bring my, yes I did bring it. Let's hope it has power. So one of the basic things that's going to be happening in regular sequencing, you're running it like in a gel, or you're running it in capillary electrophoresis. With next-gen sequencing, it's a standalone instrument that uses some oligonucleotides that are designed to bind to other specific sequences, and I'll go into those in a moment. And those are attached to like something that looks like a microscope slide. It's about that big, and you put it into a machine, and that thing that looks kind of like a big microscope slide actually has tens and tens of thousands of little stretches of DNA attached to it to bind incoming DNA fragments. So you might think, how many little places on a microscope slide can you bind? Many, because we're talking microscopic level of detail. So combined with this microscopic level of detail on this specially prepared slide, you also have a very high pixel number camera. And that very high pixel number camera is able to detect light down to basically single tiny little areas, not necessarily one base, because that would be more an electron microscope, but every tiny little group of bases that are isolated from a bunch of others. I'm going to explain to you how that works. Coming as a group, all putting off the same signal, it can detect that to a very high degree of accuracy. So anyhow, this is just another review. You'd have the template strand, you'd have the primer, and the incoming bases right here. And the incoming bases... Sorry, what's that? Is that someone's phone?
That was my phone, sorry.
Those incoming bases have different fluorophores attached to them. And those different fluorophores can then be detected with a laser that's on the microscope slide. So how do we get to this point? That's the big story of how do we get there. So there's a process to the sequencing. You have, let's say you want to isolate part of a genome. Let's say you want to isolate a chromosome, or you want to sequence E. coli DNA, complete chromosome. You can do an entire human genome. It's gonna take longer, but you start by fragmenting your DNA into very specific sizes, relatively short pieces. You know, couple hundred base pairs at the most, because unlike the sequencing, which some of you have seen, some of you will see, unlike that sequencing where you can get a read of 500, sometimes 600 bases off of a single primer, in this case, the next-gen sequencing is doing, millions of tiny little stretches of DNA and putting them together with a computer. So it combined the high pixel camera with the ability to put very accurate sequences onto a particular glass type microscope slide, combined with everything else, all the other processes that go into this, the laser technology for detection and everything, so that you can do this procedure. So you've fragmented your DNA, and you're basically blunting the ends and then adding an A overhang to it. And you're adding an A overhang so that you can add adapters. Adapters are an essential part of being able to do next-gen sequencing. Without adapters having specific sequences, you would be dead in the water. Because remember, one of the things you need to be able to do sequencing is you need a known sequence to bind a primer to. So if you just have all these short little random fragments of DNA, you don't know the sequences necessarily of any of your fragments. So if you can take your fragments and bind adapters to which you know the sequence inside those adapters, then you have a surface to which you can bind that primer and start sequencing the unknown section of the DNA in between the adapters. So you're adding an A overhang so that you can have an adapter just with a little T overhang and ligase to fix it. And so you're specifically selecting for that DNA. So then you're going to hybridize that to the flow cell. So the flow cell is kind of like what I showed you, that little microscope slide. And you're going to extend them, and I'll be showing you the extension and amplification process, how that works. And then you sequence one strand, and then you sequence the other strand, and then you put them together with computing power. So that's the overall picture of what's happening. So here's that little microscope slide. It has a bunch of little channels in it. You can use one channel each for a number of different sequences. Let's say you're sequencing just E. coli genome. E. coli genome is not nearly as complex as a human genome. So maybe you would just want to use one or two channels for that, and then your next sample, you could have 2 channels for your next sample, and one channel for another maybe short bacteria you wanted to get part of a sequence and stuff like that. Or if you're sequencing an entire human genome or something very large, you could use all 8 channels to make sure you're trying to capture as much of the variety of sequences as you possibly can. So you can have individual samples in those channels, or you can do same sample in all 8 so that you're getting as much overlap as possible in them. Okay, so what is on those tiny little lanes of that slide right there is, in regular formation, you have short little oligonucleotides. And the oligonucleotides have a specific sequence that are designed to bind to, my pen won't go that far accurately, designed to bind to the little adapters that you've now put on the ends of your piece of DNA that you want to get sequenced. And there are 2 sets of little adapters, one for the forward strand and one for the reverse strand, because DNA is double-stranded, so you want something that's going to bind to both strands of your DNA. So what happens is, as I've sort of said, you get your DNA fragment, you blunt it with an exonuclease. You can cut it back or you can fill it in however you want. You phosphorylate so that you can have a 5' phosphate. You add an A overhang to it, and then you're adding that adapter that has an extra T on the end right here, and ligase is making that joint. You have to phosphate, you have to add a phosphate so that you're making sure you have a phosphate for the hydroxyl group on the adapter basically to bind to. So, and you're then PCRing up, so you don't have just one copy of things, you have multiple copies of your target sequences. And so you can again use the 1 lane, or you can use the 8 lane depending on how much overlap you want. And then you're picking out a good size range that has come out of this PCR reaction. So remember, you're starting with multiple copies of your target. Let's say you have a genome. You're not starting with, say, 1 set of chromosomes. You're starting with multiple sets of chromosomes, so that when you shred those chromosomes into small fragments, maybe this copy gets this part of the chromosome and this part of the chromosome and that part of the chromosome, that in the correct size range, and then another set of oligos would have like that part, and a different part over here. And so through having multiple copies of what you start with, overall in that chosen middle of the range size range, you tend to have a genome represented in that total size range. So it's a particular piece of instrumentation. It's a piece of instrumentation that you can just pop that microscope slide into, and it has the lasers all built in. It has places for your chemicals, and actually it's a lot like kit chemistry. You pop the microscope slide in, you pop all the chemicals in that they're going to use, and the machine is doing the rest and sending data to the computer. But here's what it kind of actually looks like. There's that microscope slide flow cell right here, and you've got your DNA that you've chosen from that middle range that you're putting in here, and the machine's doing all the work for you. So you don't have to worry about adding it to the microscope slide yourself and flowing it across or anything like that. The machine picks it out of there and flows it across for you and does everything properly. So that's the first thing you're doing when you pop it into the machine. You put that segment of DNA that you've chosen into like a little test tube, clamp the top on with a little like hose going to it basically, and the machine pumps the liquid across the microscope slide so it can bind to the slide. Okay, so you have on that microscope slide millions of possible places for it to bind. And if you were doing this, you choose the concentration of DNA that you're putting on that slide carefully so that you're not getting DNA fragments bound to every single one of these initially. You're doing it so a DNA fragment is binding and there's lots of space around it for the amplification process of making more copies within that small area. So here is here's the DNA section that you want to sequence, and there's an adapter on one side and there's an adapter on the other side. And the adapter on this side is binding to this oligo, which is covalently linked to the microscope slide, so it's not going to come loose. This is just a hydrogen bond right here, and so it can come loose very easily. So the first thing you need to do is add polymerase to extend this sequence to the other end. So this is your original strand right here. This over here is what's attached to the microscope slide, 5' to 3', and you need to do a 5' to 3' extension all the way along here to make it double-stranded. Because then the very next thing you're going to do, after you have the newly synthesized strand, you're going to discard this original strand. So now you have that strand that you've synthesized on this side, which is covalently attached to the microscope slide at the bottom. So it's really firmly attached. Okay, so you're going to next do what's called cluster generation. So you ideally want single molecules bound, say, in the middle of each of these little dots. And these dots would just represent where a single molecule ends up binding. It's not like you have a template that says, okay, here's a dot, there must be DNA attached here. It's, where does DNA on average attach? And you need space around each one. If you get overlapping ones like here, you probably have two different sequences right there. And the computer's not going to be able to distinguish those two different sequences because they're going to be putting out fluorescence, at two different bases, and the computer's not going to be able to distinguish which base goes to which strand. So you want them all to be very well separated from one another on this microscope slide. So you've made this, you've extended, and you've gotten rid of the original strand. And then it flips over and anneals. So the ones, because remember I said there's two sets of oligos right here. So this set of oligo is what it's binding to, and this set of oligo next to it is what is designed to bind to the adapter on the other strand. So now it's able to flip over, and this end of it is able to bind to the other adapter. And then again, you're using polymerase to extend 5' to 3'. So now you have, again, double-stranded DNA. So you have double-stranded DNA like that, and then you are basically denaturing them. So now you have two strands of DNA, one sequence here, and the opposite sequence here. So now you have both forward and reverse sequences right next to each other with the same, annealed the sequence in the middle. So this is not just a random sequence here and a random sequence here. They would anneal to each other if you let them. Okay, so then they're going to do it again. So one's near them, they're gonna go over to the other ones near them and do that same process of becoming double-stranded. And they fill up an entire tiny little area on the microscope slide, doing this many times. So at the end, you have a whole bunch of forward sequences right next to a whole bunch of reverse sequences, and then you say, okay, let's sequence the forward strand first, and we have a cleavage point in each oligo. There's a specific cleavage point that goes with each set of oligos. So let's cleave all of the reverse strands. So now we have only the forward strands left. Doesn't really matter which order you do it in, but you're gonna sequence first all the forwards and then all the reverses, or vice versa. So now you have a cluster with only the forward strands. And again, this is where those essential adapters are so important. So you have all forward strands, and then you have a known sequence, again, down here from the adapter, to which you can bind a sequencing primer. And then it becomes much more like the Sanger dideoxy sequencing that we talked about or will talk about in lab is that you have a specific primer that's going to bind to a specifically designed place on the DNA, and the polymerase is able to add bases one at a time, and you're able to see what sequence it's adding. Now, the strong point is every single one of these is exactly the same sequence. So you're not trying to sequence multiple different things in a small area. Everything in one of those tiny little dot areas is exactly the same sequence. So what happens is this pseudo Sanger dideoxy sequencing is that you have the sequencing primer binding, and if this is an A right here, you need to add a T to it. So the polymerase adds a T right here. Now, remember I showed you that it has an extra fluorophore on the end, and that extra fluorophore stops the sequencing from continuing anymore. So it's only able to add a single base before it's stopped, much like in the Sanger dideoxy sequencing, although this is done with regular deoxynucleotides. But the point here is there is something there that stops it after only a single addition so that the sequence can be determined after that one base is added. So that T is added right here, and then a laser light is shown upon it, and the ones that are all the ones that are T's in a particular dot say, I'm red, and red can be detected. Maybe a different dot nearby says, I'm yellow, because it's a different part of the sequence. So all these little dots on this big microscope slide are going to have a different sequence in every single one of those little dots, and that's why you need computing power to remember the sequence in each of those little dots as it scans over it each time and put it all together in the computer. So after you've done this with your laser, then you're able to cut off the blocking area, and then it becomes a regular nucleotide. And then it happens again. Okay, so then this is a C right here, and if it's a C, we need to add a G to it, and so then G comes in right here with its little green fluorophore, and it continues. So the laser would detect T, and then G, and then C, and then G, etc. And that is the sequence of all of these multiple strands all together in this very small area on this microscope slide. So this is kind of what it looks like in the cluster. And so this is a picture of a really high-powered microscope on the left here. Great time to cut out. Okay, a microscope going down the microscope slide with the laser, and so it's reading all the different colors and all the different dots. So 150 million clusters, 150 different sequences able to be analyzed at a single time within those 8 channels of the flow cell. So with 150 million of different clusters, some of them are going to overlap. You know, like maybe this one's overlapping with this one, and great, it happens to be the same sequence, but yellow here is overlapping with green, and maybe that's going to confuse the computer a little bit. So the computer says, no, this is too close. We're gonna rule that one out. So, but overall, with the 150 million, you have a bunch that are well isolated here and here and here and here, isolated enough here, 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 here. And so all the isolated ones, the computer has its algorithms, and it says, is this able to be determined with a high degree of accuracy? Is this not able to be determined, this particular dot, with a high degree of accuracy? So probably right here, it's not going to be able to determine those with a high degree of accuracy, maybe because the yellow looks like it's really on top of the red. That's a place where it's bound too closely. But then there's a lot of places on that microscope slide, 150 million different places on the microscope slide that you can get good sequence from. So, oops. Okay, so then a computer is able to put all those sequences together and say, you know, okay, so this one is, you know, GAATTCAG, and somewhere on that microscope slide, there's going to be a continuation. There's either going to be the opposite sequence that helps bridge from the sequence. So it can be sequencing, say 50 bases here, and then maybe there's a sequence here, but maybe on the opposite strand, there's something that goes right here. And the computer puts them all together. So the computer says, well, all these sequences match up here, and all these sequences match up here, so we know that it goes boom, boom, boom in terms of the sequence, and the computer can say, which one's the template, which one's the opposite strand, etc., and put it all together and give you one long big sequence at the end. So this is the idea of next-gen sequencing. And the goal with next-gen sequencing is to be able to determine some disease states for under $1,000. They can do it for less than $1,000 per test now, but it's the analysis that's still costing a lot of money and making the total cost be over $1,000 still for analysis. But companies like Illumina, and this is the method that Illumina uses, they are the leader in the market for next-gen sequencing. If you've ever gotten a test at 23andMe, for example, they would have used an Illumina type system for looking at all their single nucleotide polymorphisms, those single mutations in their DNA that can help line up ancestry or tell you whether you have a particular disease state and things like that. But they're just looking for very specific tiny mutations in there. So there's a little bit different than that, but it's that same idea is that you can use a piece of equipment like that to be able to look at someone's entire genetic pattern. If doctors suspect that you have a genetic malady somewhere, they can write a prescription, much like they say, oh, you're going to need an MRI or you're going to need a CAT scan. The ultimate goal is that a doctor will be able to say, well, you're gonna need to get your genome sequenced. And they are starting to do that regularly. You have a child who comes in, maybe presenting, maybe they have nausea, their growth is not normal, maybe it's connective tissue problems, whatever. They want to diagnose what's wrong with them. So right now they will start sequencing the genome to see, are there any deletions in the genome that would be abnormal and we can use to treat this. If we know what's missing, we can then diagnose and treat this disease. So that's what's starting to happen in medicine more and more. It's still early stages, but it's starting to happen more and more. Now, not every company does it the exact same way. This is the Illumina method, and as I said, Illumina has a very high market share, so most of the people do use the Illumina method, but there are other companies who do it slightly differently. Lichor fixes the polymerase and has the DNA going through the polymerase in a different pattern. Other companies, they will fix little beads to the slide, and these little beads that are fixed to the slide, you can then fix your DNA to the beads, and it's the beads that are holding it in place and doing that same type of process as up here, but around each little bead, and then at the end they fix the bead to the microscope slide and sequence each little bead. So it's slightly different procedures because one company comes up with a methodology and another company says we want to do the same thing, but there's a patent on it, so we need to do it as similar as we can for success, but we can't do it identically because then we'll be violating the patent and have to pay a whole bunch of money. So they will come up with very similar things to do. But I chose to show you the Illumina method because, as I said, they have the vast majority of market share. Okay, so this is an example of how the reads are going. So you would have, even over, say, a short sequence, you'd have a whole bunch of little partial sequences right here from all these, 50 to 100 base reads, and then the computer can put it all together. The old method of sequencing, using traditional Sanger dideoxy sequencing, when they were going to originally sequence the human genome, they were using using a stepwise method. So they would start at one end of the chromosome, for example, and they would sequence maybe 600, 700, trying to get as long as a read as they could. They'd sequence part of the chromosome, and then they would design a primer. So it was taking time because they'd have to wait a day and wait for the new primer to come in, and they'd make a primer that would bind, say, right here. And then that primer could then extend the sequence here. They'd make a new primer and extend the sequence here. So they were taking a long time to come up with the human genome sequence using this methodology. And then some very intelligent people came along and said, this is bullshit. There's a better way to do it. And with the computing power that was really coming online and high pixel cameras that had recently become more popular. As an example, in your iPhone, those of you who have iPhones and even those of you who don't, probably have, I don't know, very high pixel cameras. When John Love came here, one of the things he requested for his lab as part of his startup package, he said, I'm going to need a good camera to document my lab work. And I think they gave him a 5 megapixel camera, and his 5 megapixel camera was like $1,500 for 5 megapixels. And so that was the state of the technology before there were really high-powered cameras. So that's why they had to start it out this way when they were starting it. And then pretty soon technology overtook them. Somebody came along and said, hey, if we combine this and this and this, there's a better way, using the computing power, which had someone's computer law is that RAM doubles every 18 months, and it really was for a long time. Maybe a little more, a little less now, I don't know, but with increased RAM, better computers, and better cameras, this all became possible to make a consensus sequence of a chromosome, a genome, whatever people want. People like Forrest Rower in biology, they will isolate critters, phages from seawater and try to say, okay, what kind of DNA? Is it one phage, multiple phages? So in that case, they're not just sequencing one genome, they're sequencing multiple genomes all at the same time, and this is all possible with next-gen sequencing. I think this is the last slide. Oh, no, just sort of showing you the different levels of equipment that's available, and this is not, it's not like it's going to be a test or anything like that, just out of sheer interest, you can have really high-end equipment that is, that can take a couple days to sequence a genome, so it might take 3 days to sequence a genome versus, as little as 5 hours to sequence a genome if you need just a shorter read. If you say, okay, we think there's a deletion in chromosome 5, you're just really focused on amplifying for chromosome 5, you're not going to need as much computing power, you're not going to need as much time. But if you're doing whole genomes and you need terabyte level of knowledge, that's when you're doing like entire population genome sequences like Dr. Rower over in biology would do. So anyhow, there's different levels of equipment depending on people's needs in research. A doctor's office might have something more like this. They have the MiSeqs.